So transcriptional control involves binding regulatory proteins to the DNA. If you can have a piece of, so back to this model, so I'm glad I brought it. If you can have something that will help bind the polymerase, you have that bind to the DNA and that recruits the polymerase, that will make it go faster. At the same point, if you have something else that it might prevent the RNA from binding. It might prevent it from binding. You have what are known as repressors and activators. Okay, so get this. A repressor represses transcription. I know. A repressor prevents the polymerase from binding. This table is my coding sequence. I'm sorry, this table is my promoter. I need to get the polymerase to bind to it. I don't know. Here's the polymerase. Polymerase should normally bind to the promoter and transcribe. Make sense? If there's a repressor, the repressor will lay on top of the coding region, preventing the polymerase from binding. Does that make sense? If the polymerase can't bind, you can't make the gene. Right. The flip side of that's an activator. An activator is a protein that will help the promoter bind. So if this is a promoter, this is a polymerase, and it needs to bind, I can get it to bind that time. But if sometimes it goes right off the edge. You need something to help it get attached. Does that make sense? So if I'm bound to the DNA, catch the promoter, catch the polymerase, bind the polymerase to the DNA. It's that guy at the airport who directs the plane to land. I was going to go with the guy with little batons. Yeah. Right, the guy with the baton. His technical title. When you go to apply for that job, guy with batons. So you got a repressor that slows down transcription because it prevents the polymerase from binding. And you've got an activator that speeds up transcription by helping a polymerase bind. Interacting with these are what are known as small effector molecules. They're going to speed things up or slow things down by acting on the molecules that act on the DNA. All right, that's a lot. What they do is they make the molecule that's, say, laying down here, blocking this, more effective. I don't know, they give it a little shield type thing. This is actually fairly comfortable. They can act to change the shape of an activator, making it more likely to bind to the DNA. They could change the shape of a repressor, making it less likely to bind to the DNA, and thus speeding up transcription. So let me give you a few scenarios and see what you think. If I was to have an activator bind to the DNA, what happens to the transcription rate? It goes up. So what happens to, say, the reaction rate of the enzyme it controls? It increases. If I was to have a repressor, what would happen? It would go down, and thus the reaction rate would go down. If I had an activator that had a small effector molecule that made it more likely to bind, what would happen to the reaction rate? It goes super fast now. What if I had a repressor with a small effector molecule that made it less likely to bind? So now the repressor is not able to bind. So what happens to the reaction rate? Keep thinking. Right, the repressor cannot bind now. So it keeps going. Normal speed. Now, you are producing activators, repressors, and small effector molecules all the time, which means that your genes are being controlled very precisely in how much or how little gene product they're producing.
By big ups, big downs, little ups, little downs.
Big up, exactly.
It's like the cheat codes to old video games.
Up, up, down, down, left, right, BA, select, start. Right. So, right, I got that Konami code. I actually, fun story, I made that the extra credit on one of my exams one year. It's like, in order to get through this exam, like, with an additional 10, increase your letter grade by 1 letter grade, tell me what the Konami code is for, I think it was for Contra. Yeah, one guy got it right, and I was like, I will never do this again. I didn't expect many people to get that. That's an old. Right. All right, so bacteria produce what's called polycistronic RNA. Here's the deal. Bacteria don't produce just one gene at a time. They produce a whole bunch of genes all at once in what's known as an operon. No, I'm not going to make you do an example of an operon. I'm going to say what an operon is. Right. So an operon is a group of 2 or more genes under the control of a single promoter. So instead of, here's a promoter, haha, you got 3 genes involved now. That's it. And that's an operon. You've got many genes controlled by one promoter. It's 2 or more genes controlled by one promoter. Yeah, thanks. So what ends up happening is you've got a polymerase will bind. It's going to run the length of the DNA, and produce one super long strand of DNA. One super long strand of RNA. So far, so good? This has 3 genes on it. These 3 genes, these 3 pieces of RNA can be encoded all at once into one super long polypeptide at the ribosome. But after that, they're not one unit, they're 3 units. So they get cut. What's it called when you break a polypeptide? When you cut a polypeptide. Let's see. It starts with P. Okay. Somebody say lysing, I heard? Splicing. Sort of like lysing, but it's with a protein. So it's called proteolysis. So we have proteolysis, where you're cutting that single polypeptide into multiple units. These can then go on and connect to each other. Typically, this happens when you're trying to transcribe multiple genes that have the same functional goal. You're breaking down sugar, something like that. And then these 3 gene products, that's exactly it. Quaternary structure. These 3 gene products can then go on to do something together. Sort of like if you were to break down, okay, let me put it into more normal terms. All right, let's say you wanted to study for an exam, hypothetically speaking. One of you is really good at taking notes. One of you actually understands the material. One of you is paying for dinner. So far, so good? The 3 of you together are a functional unit that's able to study and stay fed.
You can be one adult.
Right. 3 different things that have to come together toward one goal. It's easier to produce all 3 of them at the same time than to produce them all separately. In order for an operon to work, usually you have a gene that's being expressed all the time, and it's going to stop it from working. How would you stop this gene from being, how would you stop this RNA from landing on that with the repressor, as per said slide. You're producing constantly something that's just going to sit here, stopping that from binding. Eventually, that's going to be removed. Typically, that'll get removed by whatever molecule that you are trying to break down. So the operon has a promoter region, a terminator region, promoter, coding region, terminator region, just like any other gene. Typically, it'll also have what's known as an operator site, a place for that inhibitor to bind, that repressor to bind. So you've got a promoter, you've got a terminator on the other end, and you've got your gene sequence, and inside that gene sequence you'll have an operator site where you'll be able to recruit factors that will speed up or slow this down. The promoter and the terminator you guys should get. The promoter initiates transcription, the terminator ends it. You have a cap site, which is where you're going to have a catabolic activator protein that's going to be recognized to make this go faster. It's going to recruit the polymerase. You're going to have a site where there's negative control. What does negative control do? Stops it, slows it down. So you're going to have a sequence there that a repressor can bind to slow everything down. And then, of course, you have your gene of interest right in the middle, your coding sequence. Glow-in-the-dark mice, because when in doubt, glow-in-the-dark mice.